So I will edit out the initial part. So once again, let's start. So welcome to one more show. Today, the discussion is going to be about two studies. Those studies are actually contradictory to each other. I want to see them both with you. The basic gist of the study is the following. The authors of one of the studies looked at the patients who are severely ill with SARS-CoV-2, and they found out that in these patients, the previous coronaviruses and the human coronaviruses antibodies were more in quantity, while the SARS-CoV-2 antibodies were less in quantity. So they provided a mechanism for why this may happen. And they also saw that if the previous coronavirus antibodies are more, then the new coronavirus or the SARS-CoV-2 antibodies are less because the immune system is suppressed by the previous ones. And they're calling it a back boost. And this is also called original antigenic sin. This is a mechanism known for HIV. This is a mechanism known for dengue virus. This is a mechanism known for hepatitis as well, influenza as well. And now it seems that the same mechanism is involved in the SARS-CoV-2 infections as well. So that is the basic discussion. Original antigenic sin is what we are going to talk about today. So here is Drbeen.com. This is the first study. It is a preprint. It is a small study, and they want people to do more studies around this or on this concept. And this study, I'll show you the PDF for this one as well. The contradictory study is this one. This is October 14. This study is October 12. So these are really recent studies. This study actually says the other way. It says that the patients who have milder or lesser symptoms actually have more antibodies from the previous coronavirus that are providing cross-reactivity and providing protection. So on one end, we are talking about cross-reactivity. On the other end, we are talking about antigenic sin, original antigenic sin. So we're going to compare these two mechanisms as well and try to make sense of why this would happen in some patients, as why this would cause in some patients to be less severe symptoms and in the other patients to be more severe symptoms. So that is the basic discussion that we're going to talk about. And there is a comment about the long haulers. I'm going to do a discussion about some of the long hauler mechanisms that are being reported now, hopefully on this Saturday, and then continue that with the next week. All right, so let's see. This is the Wikipedia article about the original antigenic sin. This is the Wikipedia article about cross-reactivity. And then this is another article, rather old, it's 2015, a simple mechanistic explanation for original antigenic sin and its alleviation by adjuvants. So this is the basic discussion. Let's start. So here is what the author saw. So I'm going to provide you the data or the demonstration or explanation for the study where they said that the presence of the antibodies or immune response to the previous coronaviruses caused severe illness. So let's see that. So the original antigenic sin may be causing severe COVID-19. That is this takeaway from this preprint. What they found was that in some patients who are severely ill, in them, there are more antibodies that are geared, that are targeted towards the older coronavirus antigens. And this actually causes an issue for this patient, and we'll see the mechanism. And the patient's own new coronavirus, the SARS-CoV-2 infection, does have protection as well, but that protection is less because of the previous cells that are present. So let's see how does that work. So this is the study. What they saw was 17 hospitalized ICU patients are the ones that they looked at. What they did was they kept taking their blood samples. From the blood samples, they would take the B cells. And we know that the B cells are responsible for making antibodies. They will take the B cell and then they would see what kind of B cells are these? What kind of antibodies are these B cells making? And they saw, so if you see here on the left side on your screen, these B cells a big army of these B cells. These are also called clones. Why are they called B cell clone? Because once we have a B cell that would react to an antigen, then that B cells copies will be made so that we have thousands and millions of B cells, which will all make antibodies. So there is a big clonal expansion of the B cells in these patients to the previous SARS-CoV infections, or you can say previous human coronavirus infections as well. And these B cells are making a lots of antibodies, and these antibodies are useless for SARS-CoV-2 because they cannot neutralize the SARS-CoV-2. They can neutralize human coronavirus, they can neutralize the SARS-CoV, they can neutralize the MERS-CoV, but they cannot neutralize the SARS-CoV-2. So they are really not causing any protection, but they are present in a large quantity, which has a negative effect on the patient. And again, why is that negative effect? We'll talk about that today. On this side of the diagram, what you're seeing is that these are the B cells that are for SARS-CoV-2, the current infection in that patient, but their function is really low because these cells that have proliferated before, they have suppressed this set of cells. So previous infections memory cells come into play quickly, they start functioning, and they make the new memory cells or new naive B cells that need to become mature and need to protect us. The older cells are now causing the newer cells to be suppressed. So we do not have a decent response to SARS-CoV-2. And again, why is that? Let's see. So here is the explanation for the original antigenic sin. Now, play along with me for a second. Imagine I have human coronavirus, and that human coronavirus is here. This is not SARS-CoV-2. This is a previous kind of, for example, HKU coronavirus or SARS-CoV-1 or MERS-CoV, some previous coronavirus. If you pay attention to the spike proteins that I've made here, you would see that I have marked a lot of pieces of this protein as S1. Again, these are just the names I gave. So S1 doesn't mean anything but then an antigen on a previous human version of coronavirus. This antigen, S1, causes our innate system to become activated, as we have discussed many times. So again, we are not talking about SARS-CoV-2. So once the innate arm becomes activated, that arm will make the adaptive arm to become activated as well, normal regular immune response. So at some point then, the B cells will become proliferated and they will become plasma cells and they would start making antibodies. So imagine that this happened to me 6 months ago. Nothing big, some mild symptoms, had the infection, immune system responded, B cells started making antibodies, all good. Now remember that these B cells are going to become memory B cells as well. So that in the future, if the same coronavirus appears again, they are going to rapidly react and cause protection. Normally, this whole system takes anywhere from 10 to 14 days to become active. However, once we have the memory cells, they would become active within a day or so. So now we have memory of the older infection. Then imagine that the next time around, maybe now, I get infected with SARS-CoV-2. And here, this is SARS-CoV-2 now. And if you see here, so here is the delicate point to understand. In the SARS-CoV-2, imagine that SARS-CoV has arrived with its own antigens that are different from the SARS-CoV-1 or the human coronavirus. And so the predominant type of antigens are, let's call them S2, another type. However, the SARS-CoV-2 also have a very tiny amount of antigens that look like previous coronavirus. So here, if you see, this is the S1-like antigen present on SARS-CoV-2. But now in this virus, in the SARS-CoV-2, imagine that this antigen is lesser in number. It is not the dominant antigen. In the previous infection, the S1 antigen was the dominant antigen. In the new virus, the S1 is a less dominant antigen and other antigens are dominant. However, now think about it with me. So this is the point to think about together. If our immune system is going to respond to S2, that is the dominant new type of antigen, then our immune system is going to need 14 days, 10 to 14 days to become active, because it is going to recognize the antigen, it is going to digest the pathogen, it is going to present the antigen to the naive cell. Then naive cell, in the presence of interleukin 14, will go T helper 2 route. Then T helper 2 would release interleukin 4 and 5. That would cause the B cell to become plasma cell, and then plasma cell will make antibodies. This would take time, correct? Correct. So hold on there with me. Hold this thought. New virus came in, has new set of antigens. Our system is now getting ready to respond to it, and it would take 7 to 14 days, correct? Meanwhile, if you see down here in the diagram, the previous infections memory B cells are still present there, and they're sleeping, and they're saying, okay, do I have any antigen present again? These are the memory B cells from the last infection. When these B cells encounter this S1 antigen present on SARS-CoV-2, but in lesser quantity, they still become active. That is their function. And they will become active, the memory B cells to previous infection will become active within 24 hours. Right? Delicate point here. So as soon as they become active within hours or days, they will rapidly undergo clonal expansion. That means their copies will be made, and they would start producing antibodies. And they will start producing tons of antibodies. So now the immune system is still gearing up to respond to new antigens on the SARS-CoV-2, while some smaller number of antigens have already triggered the previous previous memory B cells, and they have started producing antibodies. Similarly, the memory T cell from the last infection have also become triggered in the same way. So now think about what is happening. I got infected by SARS-CoV-2 now. My body is going to respond to the antigens for SARS-CoV-2 in 7 to 10 days, 14 days. But my previous infections, memory B cells are sitting here. They looked at some small number of S1 antigen on SARS-CoV-2, and they rapidly started responding. Here is the problem now. These antibodies that are produced rapidly are not neutralizing for SARS-CoV-2. They are really not against SARS-CoV-2 in a good way. And SARS-CoV-2 is now running around in the body. Body is producing a lots of antibodies. And you know that when lots of antibodies are produced, what happens is that those antibodies are going to try to find the pathogen to attach with. But we don't have a lots of antigen on SARS-CoV-2 that would need this antibody. So these antibodies are now running around in the body. They are getting connected. So let me show you here. Number one, these antibodies will connect with the innate system. These antibodies would loosely connect with SARS-CoV-2. They would not really neutralize it because they are really not against the SARS-CoV-2. They are just one small part of SARS-CoV-2, not neutralizing it. So when they are bound here, as we discussed a couple of days ago as well, this would cause antigen-dependent or antibody-dependent enhancement. Not antigen, antibody-dependent enhancement. What does that mean? That means this complex now of new SARS-CoV-2, the new virus, and the antibody to the old virus, this complex is now running around. It is not able to neutralize the SARS-CoV-2, but it would still become connected with the innate arm. cells, and these cells are going to pick up this complex, and they are going to become infected because the antibody is not able to neutralize the virus. And that is antibody dependent enhancement. This can happen with the previous infections of a different strain. This can happen because of vaccination as well. So this is one mechanism of immune dysregulation that would occur. The second mechanism of immune dysregulation is that now we have the immune system that is encountering a large number of antibodies. The virus is not neutralized because these antibodies are not able to neutralize it, but there are lots of antibodies that would cause the immune system dysregulation. And the immune system would just go in a supercharged mode because it sees tons of antibodies running around. Immune system thinks that, all right, I need to activate the complement system. I need to activate the innate system. And it just starts making a flood of cytokines. In this whole process, SARS-CoV-2 is still protected. It is still not going anywhere because the antibodies are useless against that. And so result is we have severe immune system dysregulation. Overactivity of the immune system is seen. Now on the cytotoxic T cell side, this is even more interesting that these cytotoxic T cells that are the T cells for the previous coronavirus, when they become activated by the new coronavirus, but without trying to neutralize it, these T cells, cytotoxic T cell, does a very weird thing. Instead of killing the cells next to them, which is their function, they just sit around and start releasing cytokines. Meaning their primary function of killing is not activated, but their function of releasing cytokines becomes activated. Response to that, the result of that is that there is a lots and lots of cytokine. That would cause cytokine storm as well. Correct? So now what is happening in this patient? His actual good antibodies against SARS-CoV-2 are still, we are waiting on them. They would take time to be produced. Meanwhile, previous antibody and cells have become activated. They would do, number one, antibody dependent enhancement, as we saw. Number two, when these cells become activated, they would suppress the activation against SARS-CoV-2 because the innate arm is connecting with the SARS-CoV-2 through the less dominant antigens. Why is it connecting with the less dominant antigen and not more dominant antigen? Because the innate arm is trained to already recognize that antigen that is present. So it would say, fine, I have found an antigen, I'm going to work with it, but this is not a dominant antigen. I don't need to work too much. So the response to actual SARS-CoV-2 is low. Extra response to previous SARS-CoV is high, and that response is useless other than it is causing immune system dysregulation. That causes the patient to become more severely ill. So if I go back here, what are the things that are happening? ADE is happening, antibody-dependent enhancement. Complement system is becoming activated. That is actually going to cause hypercoagulability as well. Then the remaining cytokine storms are occurring. That is going to cause macrophage activation syndrome, and the patient is in ICU now. So what is the good, the good, the bad, and the ugly of this whole thing? In certain cases, this is called antigenic sin, original antigenic sin. And it is good in some cases. For example, in case of flu, it is actually good. Why? Because flu virus continues to change its antigens, or it keeps drifting away from the original version. So when the new flu virus arrives in our body, not only we start making antibodies against this, but the previous B cell memory cells would become active as well, and they would mount a response as well, which is a larger response, or which is called a back boost, and that would protect us from any previous strains of flu virus that arrive in us. So it is protective in that case. We want that. The bad dengue virus is an example, where there were 4 or 5 strains of dengue virus. If I am infected with strain 1, I would develop memory cells against that. Then let's say 2 months later, I'm infected with strain number 2. And now the previous strain's memory B cells have become overwhelmingly activated. They would not allow the new memory B cells to become activated. And I would have a cytokine storm to the previous version. So now the dengue virus will be able to evade my immune system's response. So that is the bad part. How did they try to fix that, or how are they trying to fix it? I think this is a story from 2015. So they are trying to make vaccines that target all 5 kinds of dengue viruses. So that is the bad part. And then the ugly part is with SARS-CoV-2, where if somebody was infected with human coronavirus and had the B cells against that, then the SARS-CoV-2 arrives and the previous B cells become active before the new immune system becomes active for SARS-CoV-2, the patient ends up in ICU. Does this make sense? So there is a very, very good question by Carolyn. The question is it possible that President Trump could suffer antibody-dependent enhancement due to the polyclonal antibodies cocktails? So I want to make sure that we all understand this one thing. The polyclonal or monoclonal antibodies, when infused from outside, these are not going to train our B cells to become memory B cells. These antibodies are not going to trigger the immune system of our own to react to SARS-CoV-2. Instead, these are just the antibodies given from outside against the SARS-CoV-2. So will they cause any memory B cells? No. Against SARS-CoV-2? No. So will the SARS-CoV-2, if it arrives in the body again, would that cause ADE because of the monoclonal or 2 monoclonal cocktails? No, they are not going to cause it. But what can cause it is vaccine, for example. And again, let me clarify one thing. I am not anti-vaxxer. I am actually pro-vaccination. I actually think it is harmful to be anti-vaxxer. So my discussion here, I have to present it to you even when some of the points here point towards a harm from vaccine. So I want to keep my position clear to you and then still explain this, that it is possible that if I am vaccinated today and I have developed memory B cells against SARS-CoV-2, then the actual SARS-CoV-2 arrives and the memory B cells to the vaccine react overwhelmingly, and our new immune system's new response to SARS-CoV-2 is yet not active, then that overwhelming response can cause ADE plus original antigenic sin, resulting in cytokine storm. So I hope that is clear. I'm going to answer one more question and then show you the study that is opposing to this. So there was a question here. Okay, so I think that question scrolled past. Now let's look at the second study. The second study says here, this one. In this study, their observation is that they took some patients, so 60 patients, and they measured the antibodies present in them for SARS-CoV-2 and for the previous coronaviruses. And they found, let me show you in the PDF document here, which is very interesting. They found that if I go here, they found that patients, so here, imagine that this first diagram over here, the yellow one is SARS-CoV-2 patient. And they saw that if the patient had higher antibody titers of OC43 coronavirus, another strain of coronavirus, if they had higher quantities of those antibodies, then SARS-CoV-2 related hospitalizations reduced. and the patient had milder symptoms. Similarly, if you see here, the patient has milder symptoms if HKU1 coronavirus antibodies were present. And same is the case for others as well. Now, what could be the cause of these two separate responses? In one case, we are seeing that the patient who has the antibodies have a higher or more severe response. In the other case, we are seeing the patient who has the antibodies have milder response. I think, so this is my conjecture, I think the reason for this is that in one case, there is cross-reactivity. In the other case, there is antigenic sin that is activating. What does that mean? So let me just explain that one part, and then we are done for today. So here is what that means. Imagine that we have two viruses. This is virus 1. And let's say that here is an antigen on this virus. And if we make an antibody against this part, then this virus becomes neutralized. This is A. Then we have one more virus, let's say B. That virus is slightly different from A, but still it has an antigen on it that is similar to the virus A antigen. And if you make antibodies against that, that also allows it to become neutralized. If that is the case, and if then the patient has antibodies against the previous virus, when the new virus arrives and we make the same B cells from the previous ones to make antibodies, and if these antibodies can connect to that antigen and still neutralize it, then we'll call it cross reaction, and we'll say this is cross protection. On the other hand, the antigenic sin, this is the same mechanism. The behavior of antibody is different. In the case of antigenic sin, what would happen is, imagine that the antibody is against this part of the previous virus A. And this part is still, when the antibody is produced, becomes neutralized. So this virus, this antigen can also neutralize this virus. But in the newer virus, in the newer strain of the virus, the antigen here, the one in blue, is present somewhere at a useless spot. So maybe it is present still on the spike, but slightly away from the binding region. So now that antibody is still going to connect with it, but this antibody, when it connects here, it cannot really neutralize it because it is not binding at the critical part of this new strain. And now we have an antigenic sin that is happening because now we have overwhelmingly large quantities of antigens, which are antibodies, which are doing nothing. And the more they will do nothing, the more antibodies will produce, because the virus would continue to spread and the memory B cell would keep churning out more antibodies, and they're doing nothing. So this is antigenic sin. So I believe that in one study, they observed the phenomena related to antigenic sin, and the other study observed the phenomena related to cross-reactivity. In the case of cross-reactivity, we'll have protection. In the case of antigenic sin, we will not have protection. Instead, we'll have harm. So that is the basic discussion today. I hope it is an interesting and complex phenomena. I hope that I was able to explain it. If not, my apologies. We can talk about it once more. And my apologies for the initial part as well. So please do me a favor. Please like, subscribe, and share. If you would like to donate to the work, for the work that I'm doing, there is a link in the description for donation. And one question to you, I was thinking of doing a GoFundMe for the book. So there are many folks who have actually volunteered to help write the book. However, we still need funds to be able to pay them and pay artists and others to actually put the book together. So I thought that I would discuss with you, and if you are comfortable, we can create a GoFundMe. I do not know if I would get enough support in there to be able to pay for the book writing, but that is what I was thinking. Please give me your comments. What do you think? So with this, once again, thank you very much. Please like, subscribe, and share. That is something that I think is easy to do. And then we'll see each other tomorrow. Tomorrow we have an open forum. We'll go there. So JTJ says, what's in this book? It is the coronavirus book. So we'll talk about, for example, in that book, we'll talk about the immune system's behavior. We'll talk about what coronaviruses are. We'll talk about various outbreaks of coronaviruses. We'll talk about prophylaxis. We'll talk about various treatment options, various drugs that we have talked. Do you know that we are now today is 199th video on coronavirus that I have done. So content from here will be translated in the book. I want the book to have childlike illustrations or children-like illustrations, so it is easier to digest as well, and it is going to be different from medical writing, still have good and reliable information in it. So if you like it, please tell me and we'll go from there. So thank you very much for today. See you tomorrow.